All right. I'm going to do this. All right, I think we're going to get started. Welcome, everybody, to the Biochemistry Colloquia for 2022-2023. We are excited to kick off this semester's lectures with Dr. Jean-Philippe Julien. He comes to us from the Hospital for Sick Children, which is also called Sick Kids. JP did his BS at McGill University in Canada. He then went on to do his PhD with Emile Pai at the University of Toronto. I first met JP when he moved to the Scripps Research Institute under the direction of Ian Wilson. JP's big claim to fame in Ian's lab was that he solved the first structure of an HIV envelope glycoprotein trimer. And that really revolutionized how we thought about how antibodies interact with that trimer and how fusion occurs in HIV. JP has since gone on to start his own lab in Toronto. He is a senior scientist in the Department of Molecular Medicine at the Hospital for Sick Children, as well as an associate professor in the Department of Biochemistry at the University of Toronto. In addition, he is also the Canada Research Chair in Structural Immunology. JP, we are thrilled that you could come out and join us today, and we're really looking forward to your seminar.
Thank you so much, Rob, for the very kind introduction. It's really great to visit you and your lab, and of course, everybody who's here today. Thank you for taking the time this afternoon to come and hear about our research. It's really exciting to be here. And so the story that I want to share with you over the next hour is one that relates to our attempt for the design of anti-infective and transmission-blocking malaria vaccines, and really using as a guiding principle antibody potency. And so hopefully at the end of the talk, you'll be convinced that antibody potency is a good metric to follow for vaccine design. So the topic of today, of course, is malaria, which of course is a global health priority. So malaria is not caused by viruses, it's not caused by bacteria, it's actually caused by parasites of the Plasmodium species. Half of the world at any given time is at risk of contracting malaria. That leads to about 240 million cases per year, of which over 600,000 deaths annually result. And this graph shows that in recent years, there's been an increase in the number of incidents and mortality associated with malaria, particularly because of drug resistance that has emerged. So this is very worrisome, as we had a nice declining trend for several decades. Mortality hits the hardest children under 5 years of age, and primarily in sub-Saharan Africa, as you can see here, which is disproportionately affected compared to other regions of the world. So this is the life cycle of Plasmodium parasites, and I hope that the first thing you appreciate is how complex it is. It is a mosquito-borne disease, so female Anopheles mosquito that have in their salivary glands these sporozoites are what infects humans during a blood meal that they go to take. Sporozoites are deposited in the skin in this way and then migrate through the blood vessels all the way to the liver, where they can then infect hepatocytes. Here they become intracellular, they mature, they completely change their form, their proteome, what proteins they express, and then they become merozoites when hepatocytes explode. And then they entered into this blood stage where merozoites now invade erythrocytes. And here this cycle goes on with multiple cycles of replication, and this is when you experience malaria-associated symptoms. Now, a subset of these replicating parasites actually develop into the sexual stage at this point, coming out of this life cycle, and now you have female and male gametocytes. And this is what is actually uptaken during a blood meal by another mosquito when they come to feed, And that is what becomes infectious in the mosquito midgut, where you have a fertilization event that then leads to this ookinete that then enters, forms an oocyst from which sporozoites come out and then migrate to the salivary glands and then basically continue on. So this is, a crash course into the life cycle of Plasmodium. And this becomes important because when you think about vaccine design and you have such a complex pathogen, you have to ask yourself, well, what is going to be in the vaccine? What components of all of this proteome that is encoded by over 6,000 genes do you go after for vaccine design? And so if you think about conventional vaccines and how they work, of course, you would probably want to have a component that provides immunity so that the person who gets vaccinated does not get the infection, right? So an anti-infective component to the vaccine. So this really allows us to zoom in into the sporozoite life stage. And so if we do that, this is a different depiction. You see the mosquito, the sporozoites get deposited in the dermis, migrate through blood vessels, and reach the liver where they infect hepatocytes. So key numbers associated with this stage. So how many sporozoites are there, in fact, in the salivary glands of mosquitoes? So this is really determined by region in the world, basically. So you have areas that have very high load and high transmission sites, and others that are much lower. Okay, but you can have a range between tens of thousands of sporozoites, depending also on the mosquito strain. How many actually get transmitted to establish an infection? So this is much harder to study, but from animal models, it's estimated that between 15 to 200 sporozoites actually get deposited in the skin and are responsible for the initial infection. Okay, so that's why we think about this stage as a transmission bottleneck, because you don't have so many, right? So you have an opportunity to actually neutralize at this stage before the infection establishes. Now, how long do you have for this process to take place? So to go from the skin to the first invasion of hepatocyte is between 20 and 180 minutes. Okay, so it's a very short window for the immune system to intervene. Because if you don't neutralize at this stage, of course, what I described before is that what happens next is that these infected hepatocytes will then release, each of them, tens of thousands of merozoites. And merozoites have a complete different proteome that's expressed. So if you develop an immune response against the sporozoite, it probably won't be effective against the merozoites, okay? And so basically, the challenge is to have an immune response already in place, for example, with antibodies that would be in circulation when you're exposed so that you can neutralize and inhibit this initial infection. So that's why we think that antibodies are very central to this quest of an anti-infective. So sporozoites and antibodies. So then if you're an antibody that's going to mediate inhibition, then you need to target something that's on the surface of these sporozoites. So what's on the surface? The most abundant protein is called circumsporozoite protein, or CSP. And it is actually critical, this protein, for many elements of the life cycle of the parasite. So it has been described to be involved in development in the mosquito for oocyst, also for the migration from oocyst to the salivary gland, migration in the skin, and then some studies describe activation that then allows for the invasion of the hepatocyte. So if you knock out CSP from Plasmodium, you basically stop all of these processes. Okay, so it's an essential protein for involving all these processes. Its structure, or its sequence, domain structure, is represented here. So it's a GPI anchored protein. It has an N-terminal domain, this central repeat region, and a C-terminal domain. Okay, and basically this has been known for many decades, the central role of this protein, so it's, of course, has been the target of past vaccine design efforts as well. And the first one that was conceptualized in the 80s and carried forward in the 90s is this RTSS vaccine, which is basically taking some part of the central region, the whole C-terminus, and have a genetic fusion to the hepatitis B surface antigen that allows for its multivalent display as a subunit immunogen together with free components of the hepatitis B surface antigen. And this immunogen didn't do so well in early development, but then GSK came along with their AS01 adjuvant, and when paired together and moved forward in a phase 3 clinical trial, what was observed is that you can achieve up to about 35% efficacy for the first year after vaccination, but that efficacy fell pretty rapidly, so you didn't have much efficacy left in the fourth year. The efficacy was actually measured in children between 5 and 17 months of age, so a pretty narrow window where they tested other pediatric groups. It didn't seem to have much higher efficacy. It actually required 4 doses to reach this efficacy, and if you didn't receive the 4th dose, then your efficacy was lower, okay? So you can imagine that in sub-Saharan Africa, such a vaccine has suboptimal characteristic. Nonetheless, there was momentum to go and test in implementation studies in 3 African countries, in Ghana, Kenya, and Malawi, whether it would be possible in the healthcare system of these countries to deploy the 4 doses of the vaccine in this population and measure the impact that it can have in the field. And what was found from these results, that I'm not going to share the details of today, but basically last October was the breakthrough announcement from the WHO that recommended this malaria vaccine for children at risk based on these efficacy numbers. So this is a major breakthrough medicine because it is the first ever vaccine against a human parasite, okay? So this had never been done before, so major breakthrough, but I hope you can agree with me that these type of numbers and type of vaccine could be improved quite a bit. And so that is where a lot of our efforts have been into how can we design the next generation vaccine. So the research questions that we asked are simple. What are the characteristics of inhibitory antibodies that target CSP? And once we have this information, can we leverage it to design next generation malaria vaccine that are anti-infective? Okay? So I'll walk you through some of our findings over recent years. So here, I go back a little bit in history again in the early 1980s, work that was pioneered primarily by Ruth Nussenzweig and colleagues that looked at a monoclonal antibody, 3D11, that was actually elicited not against Plasmodium falciparum, which is responsible for most of the incidence and mortality worldwide, but Plasmodium berghei, which is a model parasite that infects mice. Okay, so sometimes you work with these model organisms. 3D11 recognized this Plasmodium berghei CSP, and it was shown in vitro and in vivo that it was capable of inhibiting sporocytes. And one of the classic picture of the native sporocyte is shown here. And what happened is that when you added 3D11 to sporocytes, you saw this proteinaceous kind of, precipitate that appeared on the surface. And so it seemed that these antibodies against CSP made CSP precipitate from the surface, and as the sporocytes were migrating forward, a lot of that precipitate basically became shed at the posterior of the sporocytes. Now, the sporocytes would make more CSP, but then if you had enough 3D11 around, more shedding would happen, and sporocytes would become immotile. Okay? So this is a very unusual proposed mechanism of how an antibody can inhibit a pathogen. So we're very far from receptor interactions and so on. At least at the moment, that is how some are thinking in the field, that these antibodies against CSP are working. So this was kind of the understanding of 3D11, but we didn't really understand at the molecular level how it worked. So we went back to this antibody 3D11. This is the P. Bergii CSP, N-terminus, C-terminus. And then the central repeat region, as you can see, it's color-coded, has 4 amino acids that get repeated over and over again. And so these are different type of motifs. And 3D11, we showed, basically bound to these peptides that are repeated here in the center. And so here you can see, nanomolar binding affinity. We went on to solve the structure of 3D11, which is shown in gray, and the different peptides that are shown here, that are kind of shown here as a stick representation in the middle. And we understood that this antibody could bind to the different motifs, even though they had amino acid substitution, alanine, proline, and so on, because these amino acids were pointing outwards of the paratope, so it could be cross-reactive. But of course, there's many of these repeats that are present in the middle. So the epitope is repeated over and over again for these antibodies. And so when we incubated with the whole recombinant protein, we saw many FABs that were clustering around this central region that's shown in yellow and was actually recognizing the epitope multiple times because there's multiple epitopes. And here you can see how from cryo-EM we found that this is the peptide that was being bound, but this Fab could bind the next motif of that peptide and so on and so forth. So you had this clustering of antibodies that was happening around the center of this protein because the epitope was repeated over and over. Now, of course, this is P. Bergii. This is the model organism. So what happens for other Plasmodium species and CSP that are relevant to human disease and malaria? So the predominant one, of course, is Plasmodium falciparum. And here you can see that this also has repeats in the middle of the protein, but they're slightly different amino acid composition, as you can see here, but still these 4 amino acid kind of repetitive units that happen over and over again. So here we wanted to go much deeper and try to understand the inhibitory response from antibodies. And for this, we collaborated with wonderful colleague at the German Cancer Research Center, Hedda Waterman, and look at 2 populations. On the left, you have 80 healthy adults that live in the malaria endemic area of Lambaréné, Gabon. And we isolated B cells, memory B cells, that were CSP reactive. And what we found is that the ability of each antibody, that you see here as a dot, each monoclonal antibody, to inhibit sporozoites was directly related to their ability to bind to this repeat region. Okay? So the more they bound to the repeat region, the better they were able to inhibit sporozoites. In a different setting, in a phase 1 clinical trial, 8 donors that participated in the trial who received live sporozoites while they were undertaking chloroquine chemoprophylaxis. They mounted an antibody response. We characterized it again, isolating 422 monoclonal antibodies and characterizing the same thing. Does it bind to the central repeat region? Yes. With high affinity? Yes. Those are the best ones at inhibiting sporozoites. All right? So this central repeat region is really the focus of highly inhibitory antibodies in the context of Plasmodium falciparum as well. We also look, and I don't have time to go into details too much about this, but antibodies that target the N-terminus or the C-terminus, and found that these monoclonal antibodies basically don't have the ability to inhibit sporozoites, even though they bind to the recombinant protein. And same thing for the ones that target the C-terminus of CSP. They aren't able to protect against peristemia in a mouse model. Okay, so these regions are not, to our knowledge, associated with high antibody inhibition. At least we haven't found any antibodies that do so yet. So the central repeat region is really where inhibition is coming from. So if we look at falciparum, here you can see that they're color-coded. The majority of the repeats are made of this N, A, and P amino acids. All right. You have a few repeats that are NVDP, so very similar, just a bulkier hydrophobic side chain, a negative charge here that appears, and a single NPDP motif at the beginning. But very, very, very similar, right, in terms of motifs. And if we think about it from a structural perspective, what does a repeating sequence like this look like? So if we do molecular dynamics simulation, this was done by Regis Pomes, we basically find that it's a soup of spaghetti, very low secondary structure propensity. Sometimes you'll pick up type 1 beta turn that appears, but mostly we think of this central region of very low secondary structure propensity. Now this is how it is on the surface, we think, of the sporozoites, but if you have an antibody that comes in and binds, at least in the monoclonal context, what we found, this is the one antibody that can pack with the highest density. As we found, this is 19 different antibodies that bind, are clustered around this central repeat that here is shown in red, and basically induces this spiraling conformation of this otherwise low complexity structure. And so here you can see that in order to pack so densely, there are also quite a lot of contacts that happen between the different antibodies. So this is the case for falciparum. The other one that matters a lot in the field of malaria is Vivax, because Vivax has a slightly different repeating sequence, as you can see here, with these much longer repeating motifs. So Vivax affects other parts of the world, mostly Southeast Asia, South America, and Eastern Africa. And so we wanted to see if antibodies against Vivax played the same role in clustering around the repeats and being inhibitory. And I won't go through all the data, but the point is that yes, Vivax, the same thing. You have lots of antibodies that cluster around the central repeat to mediate its inhibitory function. So the same theme seemed to happen over and over again, and so we started paying closer attention to antibodies that were recruited in the response. And so you can imagine, vast diversity of antibodies that exists in the human population. We had many different individuals as well. And when we looked at the genetic signature of these antibodies, we saw that one type of gene, the VH3, was much more recruited compared to any other type of antibodies, a particular length of CDR3 in the light chain. And we wanted to understand why. And so here is the gene, that VH3, that's here. And then here is how often do you see a somatic hypermutation at that position. Somatic hypermutations happen in antibodies so that they can gain better affinity, right, to the antigen. And that's what we saw. There were some regions across humans that were being mutated in these antibodies in order to increase the affinity to the repeat, as you would expect. But what we saw is that there were other mutations that were being enriched quite heavily at other positions that didn't directly bind to the antigen. So where did they bind? They actually bound to the neighboring antibody. All right? So during the antibody response, there was maturation towards better binding of the neighboring antibody in the context of binding to this repeat region. And so this was the first time that such homotypic interactions for affinity maturation were described. And when we looked at the different studies that we did and studies that were done by Ian Wilson and Andrew Ward, in the context of the RTSS vaccine and antibodies that were elicited then, we saw that these homotypic interactions were being mediated and evolved for by the B cell during the immune response in order to improve the affinity and B cell signaling. And this was true for humans, also for mice. This was true for Berghei, Vivax, as well as Plasmodium falciparum. So we think that this is a theme across immunology that had been characterized before, that these type of responses to repeat antigens were kind of uncovering. So the way that we think about it, of course, all these studies are in the context of a monoclonal antibody, right? Now, the B cell is such a context. On the surface of a B cell, the BCR is monoclonal. You only have one antibody receptor. And so this is where the selection of mutation is made. If you have mutations that lead to higher affinity, this B cell will signal stronger and will be selected positively and will lead to clonal expansion. And so we think that this ability for homotypic interactions to develop is being selected for at the level of the B cell, and we do have some data to support this. I don't have time to go through it today. The question becomes, is having more of these homotypic interaction or more antibodies around the repeat, does that lead to better sporocyte inhibition? And so far, in our attempts to characterize this, we found that no. It doesn't seem to be a direct correlation between having more antibodies on the repeat and better inhibition. It seems that where it is being selected for is not to kill more sporocyte, it's to have more B cell activation. And so that is where the selection process happens. The second question is, does this homotypic interaction and strong B cell activation impact the durability of the immune response in vaccination? And this is a critical question because, of course, we want our vaccines to have the longest durability, and we want the B cells that are brought into the response to accumulate enough somatic hypermutation to differentiate, be of high affinity and high quality, and be brought into the memory. Now we think, and this is our working model, that in fact, if you have too strong of B cell activation at the beginning, those will lead to short-lived responses that will not be very durable. And so we actually do think that there is a role for how many repeats are in the immunogen that would impact in the durability. So this concept of homotypic interactions we think have a lot of implication for vaccine design. The second one that I want to highlight is this concept of the different motifs. So lots of NA, NP, so I said that already, but some minor repeats, and particularly those that are close to the junction with the N-terminus. So do the antibodies have a preference for one or the other, or are they cross-reactive? And when we looked at many monoclonal antibodies, we found that they are basically all flavors. Some are specific for just NAMP, some are able to recognize two motifs, and some are highly cross-reactive. And so the question became, well, what should we put in our vaccine? Which ones are the best antibodies? The ones that just target NAMP or the ones that are very cross-reactive? And so this is work that was done by Elena Levashina at the Max Planck Institute, where she took a lot of monoclonal antibodies, passively transferred them into mice, subjected the mice to mosquito bite challenge. Those mosquitoes have sporocytes in their salivary glands, and then basically 7 days post-infection, measure whether the mice are parasite-free or whether they're infected. And so whether these antibodies are protective or not. And what she found is a bunch of antibodies that did pretty well and some that didn't do so well. And when you look at the ones that did pretty well, or the best, it turns out that they do have this cross-reactivity to different parts of the repeat. And in fact, some of them can have very good inhibition or protection, like CIS43, by having more of a focus on the minor repeats or the junction compared to the NAMP. And work by Bob Cedar that recently came out showed in a phase 1 clinical trial that if you took this monoclonal antibody, Cis43, passively transferred it into humans, and did what's called a controlled human malaria infection challenge, so where you challenge people participating in this study with mosquito bites, with sporozoites, that this antibody could confer sterilizing immunity. Okay? So this was a big proof of principle that if you could have such antibodies already present in circulation at high enough level that targeted the right epitope, that you could aspire to having sterilizing immunity. So with all this knowledge in hand, we started to design a lot of immunogens, and most of them failed, as it tends to do. So this is version 211 that I'm showing to you today, where basically we said it seems that the best antibodies recognize this region, and we might not want to have too many repeats. So we actually decided to focus our immunogen on this part. And this is in contrast to the RTSS vaccine, just to remember, that has a lot of these NAMP repeats and the whole C-terminus. All right, so we are using a different portion of the CSP in our lead immunogen. The next 3 findings are the following. If we had just the peptide or the recombinant protein, it wasn't as good as an immunogen as if you made a nanocage and that you had multiple copies of this peptide displayed in this multivalent array. And so this isn't super unsurprising in a way. This has been shown across many fields that the multivalent display of an antigen can be highly beneficial to the response. And so nanocage displays what we have. The third one is a little bit more surprising. Now, if you think of viral glycoproteins that we hear a lot, of course, SARS-CoV-2 spike, RSVF, HIV envelope trimer, all of these are heavily glycosylated proteins. Now here, I didn't talk a single time about glycans. This protein is not glycosylated, especially not this region. So whatever we're presenting and the nanocage display doesn't encode for any N-link glycosylation sites. We found that if you engineer in N-link glycosylation sites on the immunogen, we actually get a much stronger response. And this is aligned with some recent studies by Daryl Irvine and Bill Schief, where they think that a lot of the immune response is mediated by these lectins or complement-mediated receptors that lead to biodistribution of the immunogens. And the last thing that we optimized is around T-cell epitopes. So this region of the antigen does not contain a T-cell epitope, or not a very strong one. And so we know that to have the right type of immune response that's long-lasting, you need to have a strong T-cell immune response. So we actually included an external T-cell epitope into our immunogen. That's the lead. Okay, so all of these are important, we think, in evolving the right type of immunogen. So now we have an immunogen that only requires 2 doses, a prime and a boost, at least in mice, that can elicit very high peak antibody titers, not just to the NAMP or the repeat, but also to the junction, these minor motifs, that is durable over time. So here you can see good titers at day 40, still very good titers at day 141. The antibodies that are elicited, their quality is quite good. So you can inhibit over 80% of sporozoites and also, for a very long time. And then if you challenge these with mosquito bites, at day 51, shortly after the boost, you get sterilizing immunity, and you still get really, really good protection out to 142 days. And so we're very excited about these advances, and at least in a preclinical setting in mice, we find that this is superior to a lot of the benchmarks that we've looked at. And so we'll be excited to move forward with a partner to take this to further preclinical and towards the clinic shortly, we hope. So all that I've told you about so far relates to this part of the life cycle, right? Take a vaccine, elicit antibody immunity, have sterile immunity. That's, as good as you can get. But of course, we know that not all vaccines, of course, can confer this sterilizing immunity. And sometimes, of course, there's breakthrough infection. Now, in the context of global health, the concept of transmission is also very important. So if there is a breakthrough infection, does that mean that you're able to pass on the parasite onwards and infect members of your community, members of your family? And so we think that another opportunity for a vaccine to act as a global health tool is also to block transmission. All right? And so this is a concept that's not new to us, that's been around in the field for malaria for a long time. And so this is the second bottleneck where you could stop transmission. This is one that benefits the host. This is one that benefits more in the community setting of global health. And so let's zoom in into this transmission bottleneck. So here you have, as I said before, sexual gametocytes, male and female. 15 minutes, they come into the mosquito midgut. They're taken during a blood meal. And then 1 hour later, you have the fertilization event in a zygote. And it takes about 1 day or so for them to become ookinets that will then traverse through the midgut epithelial and establish these oocysts, for these sporozoites to then go on and be transmitted again. So we think that there's an opportunity to act at this stage, all right? And so what if there was an essential protein that existed for this fertilization event, and that we vaccinated humans against that protein, such that when the mosquito takes the blood meal that contains these gametocytes, they would also take the antibodies from the human that blocks this step from happening. And so this mosquito would never become infected and could never pass on the sporozoites because they would never develop in the first place, all right? And so this concept is called a transmission blocking vaccine. So again, being guided by antibody research, the first question is, does such an antibody exist? And if such, what is a target? And so there's been a lot of research in this field, and it was shown that indeed a monoclonal antibody existed that was transmission blocking, and what it targeted was this protein called PFS4845, and that you only needed a small amount in order to reduce transmission by 80% in a model of transmission, okay? So this PFS4845 protein, it turns out, is expressed on the surface of male gametes and is involved and critical and essential for their fertility. So an antibody that targets this antigen specifically can block this process. Now, our collaborators in the Netherlands recently entered this monoclonal antibody in a phase 1 clinical trial and have recently published very encouraging data where the antibody is safe and can stay around for a while and can keep this transmission blocking activity. Okay? Now, other projects have shown that there are other targets that are transmission blocking, like PFS25 or PFS230, but the best antibodies against those targets, turns out you require much more antibody in order to have the same transmission blocking effect. So we believe because of that, these are poor choices as the core antigen to go after, because a vaccine would need to elicit much higher antibody concentration in order to have the same effect. And so we've been chasing this PFS4845 as a top priority. So this antibody, what does it bind? So here again, we turn to structural biology, and we looked at this 4845 protein on the surface of male gametes. This is a GPI-anchored protein again. This antibody binds to the third domain. That's here, that's C-terminal. And unlike what I told you before for CSP, this antibody binds a highly conformational epitope. So you can see in orange where it binds, and here's where these orange clusters are on the primary sequence. So this is a nonlinear epitope. Here are the disulfide bonds in this protein. So you can see that the conformation is very important for this antibody to bind. So now we thought we had the blueprints to then start to engineer immunogens, and we thought conformation was central. The problem with this domain, or 4845 in general, is that there's been lots of attempts to make it recombinantly, but it's very hard to make. And if you do make it, it's very unstable, okay? And so we turn to in silico tools to try to stabilize it, with the hypothesis that if we could and can present this conformation in a better way, it would elicit better antibody responses. So here is the 6C domain, or the third domain that's shown here. And through extensive iterative processes, we found that there was 5 mutations that could be introduced in the core of the protein outside of this epitope that would stabilize the fold. So here you can see the thermal stability of the wild-type protein, and so 40 to 50 degrees. So not very good. The yields are atrocious. And this is the size exclusion profile that you get. And so we solved the structure from this black shoulder here, basically, in the previous slide. So this wasn't easy. But here, if you introduce these 5 mutations, you can see that thermal stability is increased by 25 degrees. The yields that you get now are, make the graduate students, the postdocs, much happier. And the monodispersity of the peak that you get is much better, right? We can also put this protein at 40 degrees, another temperature, for 4 weeks and run size exclusion, and it looks like that. It stays folded by CD, and so this is very robust engineering, we think. But does it matter for the immune response? So having this engineered antigen, we then collaborated with several groups. One is John Lovell with the liposomes that I'm not showing here, but here a collaboration with Neil King, where we took our engineered antigen and put it on his nanocages I-5350 that are a 2-component system. We also could put it on Helicobacter pylori ferritin, as is shown here, basically having multiple copies of the antigen. This is how the immunogens then look. And then we immunize mice. And to our surprise, what we saw is that the antibody titers that we elicited, whether it was the wild-type antigen or whether it was the engineered antigen, was basically the same. So the quantity of the antibodies that you elicit doesn't change. But when you look at the quality of the antibodies, that's where it's a complete game changer. So this is the quality of the antibody response from the wild-type antigen. So if you take the sera of the mice, you dilute it 1 in 9 or 1 in 27, you barely reach 50% transmission inhibition. But if you take these engineered antigen, now you can dilute the sera to 1 in 253, and you can get near 100% transmission reduction activity. So this is 1 to 2 orders of magnitude improvement in the quality of the antibody response that we characterize, at least in mice, from an engineered immunogen. And we're quite excited about this because, of course, antigen stabilization from structure-based principle had been described before, of course, for SARS-CoV-2, the spike, which Rob was involved in, also RSVF, and a lot of viral glycoproteins. But to our knowledge, this is the first time that it was demonstrated in the context of a parasite antigen. Of course, this is designed based on one antibody, TB31F, and it's one of the best ones. We've since gone into humans and looked at more antibodies and where it binds on this antigen, and we're excited to report that there's other sites that are also very potent and that our engineered immunogen is able to also bind. And so it's not like there's many conformation and ours is only compatible with a subset of antibodies that are inhibitory, but we think that our immunogen presents the right epitopes for a large subset of inhibitory antibodies. And I told you before that PFS230 wasn't a viable target because the antibodies that were described weren't so potent against it. But in a collaboration with Matthias Schor in the Netherlands, again, mining deeply the human antibody response, we were able to find antibodies that bound to PFS230, that bound in a distinct location compared to the ones that were described before that had much lower potency, as you can see in these graphs, that required a lot more antibody to achieve inhibition. These ones that are here and target this site actually require much lower antibody in order to reach 80% inhibition. And so we think that this knowledge now unlocks PFS230 as well as a viable immunogen for vaccine design for transmission blocking. So I hope that I convince you over the last 45 minutes or so that this approach of reverse vaccinology 2.0, which we didn't invent, but that was applied by others in the context of viral targets primarily, where you take natural responses and monoclonal antibodies that derive from these responses, whether endemic areas or phase 1, 2, 3 clinical trials, and characterize these antibodies, how they are in vitro, looking at the ones that are potent, looking at the ones that are non-potent, and validate that in vivo, understand how they bind these different antibodies, and then apply this knowledge to immunogen design, that you can come up with immunogens that would be superior to the native protein that you can then validate in preclinical models. Of course, I've shown data on CSP, 48, 45. We think now 230, we have the blueprints to start designing. And so we're interested to see how this basically approach can advance next generation candidate for malaria vaccine development. I'll leave you with two messages. So these are the things that, if you missed the previous part, the last 45 minutes, this is, the two sentiments I think that are the most important. The first one is that I've described anti-infective or transmission blocking strategy for malaria vaccine. But I think that the future is very much of a single vaccine that is multi-stage, that incorporates into a single immunogen. And so that is what we think will be beneficial and necessary, in fact, for elimination and eradication strategies. And there was a nice paper that showed, this is our work, but that basically showed that if you have 50% efficacy for an intervention at this stage, transmission after transmission cycle, you can lower initially, but then you will have everything that comes back. All the mice become infected again at 50% efficacy only at this stage. But if you combine 50% efficacy here and 50% efficacy at this stage, what you see is that you can collapse completely the number of infection through the number of transmission cycle that happens in a mouse community. Okay, so this isn't human data. This is kind of an artificial system, but that demonstrated that by targeting the two transmission bottleneck at the same time, even though you have only 50% efficacy at the two stages, you can have synergistic effects that will then allow for your vaccine or your intervention to really be transformative in a population setting. That's the first thought. The second thought is the following. So it's similar to what I showed in the previous slide. Antibody research, high-resolution structural biology, and structure-based design can lead to improved immunogens. So we knew that already. But the point that I want to make here is that I didn't tell you much about the function of CSP. What does it bind to? What's its receptor? Or 4845, what does it bind to? What's its target for the fertility? Does it have conformational changes? The truth is that the field doesn't know much about that. But the point is that even in the absence of a complete understanding of the biological function of the target, if you follow the antibodies and which ones are potent, which ones are not, you can take a pragmatic approach to already find the best antigen and the best conformation to target for design to come up with the right set of immunogen. Of course, these antibodies then become tools as well to go back to the fundamental questions that I laid out. What are the targets? What are these antibodies inhibiting? And so we are excited to also tackle these questions to see, what are the functions for these antigens that seem to play such a critical role for Plasmodium biology. So these are the two thoughts I wanted to leave you on. Of course, a lot of people in the lab that work on these projects are fantastic collaborators that I tried to mention along the way. Hedda Warderman and Elena Levashina primarily for our work on anti-infective vaccines. Matthias Shore, Neil King, John Lovell, and Kazutoyo Miura for all our transmission blocking vaccine and many others. We're so privileged and lucky to work in an environment that gives us access to such great facilities, whether they're at synchrotrons, whether they're core facilities at our own research institutes. There's a lot of study participants that partake in this research and accept and willingly donate their samples so that we can advance in this way. And so they need to be thanked at the forefront, and of course our funding partners for supporting this research. And thank you so much for your attention as well. I'll be happy to take any questions.
Thank you, JP. Do we have any questions right off the bat? Scott?
I was particularly intrigued by these kind of super antibody assemblies on the multivalent CSP repeats. And I was curious if in the context of the actual organism, when the antibodies are interaction, are those structures forming on single CSP receptors, or are they crossing multiple CSPs? And the reason I wonder that was maybe whether the rare epitope, or the rare repeats that seemed important but are kind of sprinkled in might help some sort of bridging interaction across receptors.
It's an excellent question. And the short answer, I'll give you that first, and I'll go into longer answer, is that we don't know. Okay? So it's not an easy thing to go and study from a structural perspective on the surface of sporozoites. This is largely a very small and unstructured protein on the surface of these sporozoites. So you can imagine that visualizing it in the first place is very hard. The second thing that's very hard for your question is that in the biological setting, it's a polyclonal antibody response. So you don't just have one type of antibody that can kind of cluster in this way. And of course, that's how we do our studies, right? We just incubate with one antibody and we see the spiraling. In the context of basically, the whole immune response, you have many different types of antibodies. So do you see such a spiraling effect that happens as well? So that's something we also don't know at the moment, and that we need to study more in the polyclonal setting in terms of how it interacts with the Plasmodium parasite. The third thing that I will say is that these CSP proteins on the surface of sporozoites are thought to be incredibly dense, okay? And so what happens when you take a polymer that is largely disordered and quite long, right? And you start to compress it in space. So initially you will have a lot of, radius, right, from your disordered. But then as you start compressing, then you might start to have this concept of polymer brushes that we think happen. And that can lead to actually quite a different state on the surface of sporozoites in themselves. So why are these repeats in this way? Why are they present for the sporozoite? We don't know. But one hypothesis would be that it is acting as a polymer brush to create this barrier on its surface as well, and that these antibodies are able to disrupt that barrier. And so that's one of the hypotheses that we're also studying from a biophysics and physical property perspective.
I can shout it out.
Shout it out, Anne.
A related question. All your structures necessarily are with FABs, but real antibodies are bivalent.
Yep.
And they like to bind over two-fold axes of symmetry, which you clearly don't have in your repeats. So in viruses, when that happens and you only bind monovalently, the other arm grabs an equivalent one, and that causes precipitation. So is the bivalent binding what's causing what looks like precipitation on the surface?
Yeah, it's not, you're 100% right. And it's not an easy thing to answer. What I can say is that if we do incubate, and maybe you were one of the reviewers in that paper, because they asked exactly that question. If we do incubate with IgGs instead of FABs, do we see the spiraling? So the answer to that is yes. So it's much harder to purify that complex exactly because of what you said. There's these bivalent arms, so that when we incubate it with the recombinant protein, most of it becomes in the aggregate, right? It becomes these large oligomers, right? Many IgGs, many recombinant proteins, void volume. But there is a subset that you can still purify from the main peak that is just that one spiraling complex with all, I guess, the other FAB arms that are pointing outwards. And so you get the contribution of one FAB arms, and we characterize it by EM in its supplementary figure. So that's for the recombinant system. The point that you're making is what happens on the surface of the organisms themselves, and do you get that cross-linking that you were referring to, and does that lead to the precipitation mechanism? And I think that's an important question, and I don't have much insights into that, but something to look into for sure.
JP, I have a question about sterilizing immunity in your sporozoites. So in some of your graphs, you say, we were able to neutralize 80% or 90% of the sporozoites. The ones that you didn't neutralize, right? So this, slightly sub-sterilizing immunity, does that still cause clinical infection sorts of symptoms? Is that just as bad as regular infection? So, is it just a little bit of escape from that sterilizing step suddenly becomes full-blown malaria, or do you see decreased symptoms where a lesser response might also be helpful?
Yeah, so that's a good question as well. So, when I showed in some of the graphs, 80% or 90%, again, that's a concentration dependent. So if you add more antibody in that in vitro assay, you can reach 100%. So we always try to do it at sub-stoichiometric concentration so that you can call an IC50. So that was the representation for the in vitro. For the in vivo, if you do get breakthrough infection, is the disease the same? So in our model system in mice, the mice do succumb. So it is as bad, and this is a Plasmodium berghei PF-CSP transgenic parasite line that allows for these infections to happen. So we're not sure, but we can't really tell that there's any benefit. They end up dying, the mice. The last thing I'll say on this topic is that 35% efficacy for the vaccine, RTS-AS01, is actually not protection. It's not efficacy against infection. So they didn't actually have that as an endpoint in their clinical trials. So we don't know if the vaccine actually helped to prevent infection or not. that efficacy is actually against severe disease. So it measures clinical malaria. And so the answer in the human context is yes, that the immune response elicited to the RTSS vaccine, which is CSP-based, can lead to a reduction of clinical malaria. Yeah. So it's a little bit, complex, but, so I said to your question, the answer is no first, and then I said the answer is yes second. So apologies for that. Yes?
Along a similar vein to that, has there been any interest in targeting more like midpoint stages, like the post-sporozoite, but like,
The
liver stage or
Merozoites.
whatever?
Yeah. So absolutely. This is a fantastic question too. So the blood stage is where you get clinical malaria. So why not target your vaccine against antigens there? Because maybe you still get infected, but you don't develop, disease symptoms. And maybe that's the most beneficial. So there's a lot of research that's ongoing against blood stage antigens. In our lab, we haven't started on those yet. And the reason that we didn't prioritize these targets is the following. I didn't talk much about, sequence diversity. Like we hear a lot about that for viruses, right? All the genetic mutations and the escape. And turns out that for those two bottlenecks, CSP and 48, 45, and 230, very little sequence diversity in the field. Okay, so that doesn't seem to be a topic. Those repeats, they're 100% conserved in amino acid composition. What they change is in the length of the repeats between the different strains of Plasmodium that circulate. So that's true for the transmission sites. When you look at blood stage, the antigens that are there have massive amount of diversity and antigenic variation. And so that becomes a similar question to vaccine design for those viral glycoproteins from RNA viruses, for example, that are genetically diverse, and that's difficult to vaccinate against, right? And on top of that mutation diversity, there's also a lot of redundancy in the pathways. So for one molecule that adheres to one receptor, even if you vaccinate against that antigen, there's redundancy. There's 7 other molecules that can also bind to that receptor that will, get around that immunity. So that becomes a much bigger topic for blood stage antigens. So that's the challenge there. Yeah, but ultimately, in my dream, in 20 years, maybe a multi-component vaccine against malaria also includes the best antigens against blood stages, such that you could have something like the ManB GSK vaccine that has 4 different antigens that are in one vaccine in order to deliver the best immune responses against different stages.
Cool, thank you.
Correct. Correct.
So I'm curious, is it like, does it have something to do with species recognition or something? Like why would it be so certain in one, but then to be able to diversify so much as you step outside?
Yeah, I don't know the answer to that. Why evolution did that. It could be because of what it interacts with, I don't know. But it does seem that the theme of low complexity and low secondary structure propensity is maintained across all these structures. So we're quite confident in that. So a lot of alanine-rich, glycine-rich, and proline-rich regions and repeats. So yeah, but I'm not 100% sure. The challenge there is that if you develop the best vaccine against the falciparum repeats, which that's what our goal is, it means that it won't be cross-reactive to Vivax, which is the second most responsible for second most cases, and that's a problem, right? So we do know that eventually we will have to do Vivax-specific research, but we're starting with falciparum. Yeah.
Great. Thank you so much, JP, for an outstanding seminar. Let's thank JP again.
Thank you.